Hello, myself Anushri Zogaikar. I am a student of TYBSc Microbiology, Department of Microbiology, Ruya College. And for my 5th semester assignment for paper 501, I will be talking about induced mutation and in depth about chemical mutagens which cause changes in base. For the introductory part of my topic, I will talk about mutation first. Mutation is a change in the nucleotide base pairs or nucleotide sequence which makes up the DNA. Now, there are many causes of mutations. Some are spontaneous, some are inducible, about which we will talk further in the next slide. But I want you to know that mutation is of three types majorly: physical, chemical, and transposon insertion. So, an example of physical mutations could be radiations. A very common example of a mutagen that causes harm to us is UV radiation from the sun. So the sun has UV radiations, UVA, UVB and UVC. These cause oxidative damage to our DNA. So that comes under physical. And then chemical mutagens, we have on the top of my mind, we have ETBR. Ethidium bromide, which we use very often in gel electrophoresis, agarose gel electrophoresis. So that is an intercalating agent, which means, so if your DNA is wound like this, it will have some affinity to the nitrogenous bases and it will just go in the intercalating spaces of the DNA and attach to the nitrogenous bases. This is a tumor inducing meaning it's very carcinogenic for us. Induced mutation is carried out by scientists when they want to experiment with some biological systems like organisms or plant cells. So they induce mutations purposely in the test organism so that they can study its effect after it has been phenotypically expressed. This in short is called induced mutation. The definition of induced mutation is on your screen. This slide categorizes mutations as spontaneous and inducible. Spontaneous mutations occur naturally without any need of a mutagen. This may be due to replication errors, slip strand mispairing, wobble base pairing, depurination, deamination. Deamination, we will be talking about that in chemical mutations further. And tautomerism, unequal crossing over, etc. etc. Now, inducible mutations require a mutagen. Mutagen is that agent which will induce or which will actually cause mutation, which will either modify the base pairs or insert or delete, as we will see further. Inducible mutation is further classified as chemical and physical. Physical, you have ionizing, non-ionizing radiations, UV rays, gamma rays, X-rays. Under chemical mutagenesis, we have various other mutation causing mutagens, which we will talk about in further slide. There are three major types of induced chemical mutations. One is base analog, base modifying agents, and intercalating agents like ETBR, which I have talked about in the introduction. For my topic, I have specifically chosen base modifying agents under induced chemical mutations. So I will not be talking about base analog and intercalating agents much, although there is some information in front of you which you can go through from my slide. Under base modifying agents, we again have three major chemicals which cause mutations in their own way. Base modifying agents are chemicals that modify the chemical structure of purines and pyrimidines. The modes of action are three, as I mentioned earlier: hydroxylation, deamination, and alkylation. We will talk about them in depth in the coming slides. Some prerequisite knowledge for us. So the nitrogenous bases present in DNA are categorized as purines and pyrimidines. So purines are two ringed structures, adenine and guanine, and pyrimidines are single ring structures, like thymine, uracil and cytosine. Now adenine always pairs with thymine. Thymine always pairs with adenine via double bonds. Whereas guanine and cytosine pair with each other via triple bonds. This is the basic knowledge that we need to understand the further pathways or further chemical reactions that lead to modification in the bases. Our first mode of action for today is hydroxylation by NH2OH, hydroxylamine. So this is specific to cytosine only, and we will see why ahead. So hydroxylation is basically addition of OH or hydroxyl group. Now under normal conditions, as we saw earlier, cytosine binds with guanine, right? So now when you treat cytosine with hydroxylamine or NH2OH, the NH2 group, amine group of cytosine is removed. So this process is called deamination. Deamination is basically removal of NH2 group from any structure, and hydroxylation is the addition of OH or hydroxyl group to any structure. Now this agent NH2OH causes deamination and hydroxylation simultaneously or one after the other. So cytosine, when treated with NH2OH, the NH2 group gets removed and OH group is added at the same position. Due to this, cytosine is converted now into N4-hydroxycytosine. I am extremely sorry, I have to use the word modified, not converted. So when cytosine is modified into N4-hydroxycytosine, this structure is chemically or structurally analogous to thymine. And if you remember, thymine binds with adenine. Now, N4-hydroxycytosine will bind to adenine. Upon further transcription, this adenine will bind to thymine. And this results in CGTA mutation. So if you look here, if cytosine hadn't been modified, it would have paired with guanine, which is a purine. Now, due to the transcription of adenine and thymine, thymine is being paired with adenine. So if you see, guanine is being replaced by adenine. Both are purines. That's why it is a transition mutation. Let's go to the second mode of action, which is alkylation. Alkylation, if you know, is addition of alkyl group to a chemical structure. Now our mutagen for now is methyl methane sulfonate or MMS. If you guessed correctly, methyl methane sulfonate has a methyl group. That's why under alkylation we can also say that MMS causes methylation, which means addition of a methyl group. As discussed previously, under normal conditions, guanine will bind to cytosine. But on treatment with MMS, a methylating agent, guanine is converted to O6-methyl guanine. Means the methyl group has been added at the O6 position on the guanine structure, due to which methyl guanine is structurally similar to adenine. Now adenine binds with thymine. That's why O6-methyl guanine will also bind with thymine. Now when this pair gets incorporated, further transcription causes thymine and adenine base pairs instead of guanine and cytosine. So if you look at the next slide, this mutagen is also causing a transition mutation, which is GCAT. I will explain how. So as we all know, transition mutation is when purines replace purines or pyrimidines replace pyrimidines. Now under normal conditions, if guanine had not been modified, it would have paired with cytosine. Remember, cytosine. But now it has been modified, and further transcription is causing thymine to pair with adenine. Now adenine will further pair with thymine. So cytosine is being replaced by thymine, which means these are both pyrimidines. So this is a transition mutation. Our third and last mode of action is deamination as discussed in the hydroxylation process. So deamination is removal of NH2 group from a chemical structure. Under deamination, we have 3 bases. So we will dissect these 3 reactions separately. For this deamination reaction, our chemical mutagen is HNO2 or nitrous oxide. Deamination is caused in guanine, cytosine and adenine. It is not caused in thymine. Let us look at the first reaction in deamination. So our first nucleotide base is guanine. Under normal conditions, as we should know by now, guanine pairs with cytosine. But when it undergoes treatment of nitrous acid, guanine is modified into xanthine. Xanthine luckily has the same pairing properties as guanine. That's why xanthine is paired with cytosine only. That's why there is no mutation. The second nucleotide base is cytosine. So under normal conditions, again, cytosine pairs with guanine. But when treated with nitrous acid, cytosine is converted to uracil. Uracil pairs with what? Do you know? For now I will tell the answer. Uracil pairs with adenine. So uracil will pair with adenine. And as for the replication, adenine will pair with thymine. So cytosine should have paired with guanine. But instead of that, it is being further converted to thymine. That's why it is causing transition mutation. How? Cytosine and thymine both are pyrimidines. So cytosine is being replaced by thymine. That's why it's a transition mutation. What kind of transition mutation? It is CG to TA transition. The last reaction for today is when nitrous acid reacts with adenine. So adenine under normal conditions will pair with thymine. But adenine, when undergoes nitrous acid treatment, it is converted into hypoxanthine. Hypoxanthine has similar pairing properties as guanine. That's why it pairs with cytosine. Now, if you look closely, this is also a transition mutation. How? Let's see. So, cytosine, when undergoes replication or transcription, it will pair with guanine, right? Cytosine to guanine. So, instead of adenine converted to hypoxanthine, pairing with cytosine, which pairs with guanine. So, adenine to guanine, both are purines, causes AT to GC transition mutation. That's all for today. I will post the references in the next slide. Thank you for being so patient with me. I am Anushrit Zogarekar, roll number 7511, TYBSc Microbiology, batch 2024-25. I have posted the references ahead. So for the chemical structures in deamination processes, I have used Peter Uracil iGenetics ebook, and for other chemical structures, I have used other e-sources like Science Direct and ResearchGate, which I have put the link up for. And I did some extra research and came up with some extra in-depth knowledge. So for that, I have used some other e-sources which I have linked again. For the specific bases structures, I used Byju's link. And the ATGC transition flowchart which I had mentioned, I have made it on my own. Thank you so much.